playing with that.
Yeah, you weren't here for one.
Playing music.
Okay, so anyway, so let's talk a little bit about clotting, and like I said, we'll go over the white blood cells, what they look like together, and then I'm gonna let you guys take a look at your blood slides, and we'll do differential cell counts today with the microscopes, so should be pretty quick. So the last time we were discussing white blood cells, and we talked a little bit about their characteristics. We went through the lab printout, talked about what different numbers of white blood cells could mean. If you had too much of one type or too little another type. And we did look a little bit, again, at like the morphological characteristics of the blood cells. We talked about erythropoiesis and leukopoiesis a little bit. And that's where we left off. Today we're going to look at blood clotting. Which of course is important because if you can't clot, I mean, you're in trouble. You think about how many times you get a bruise on your body and you're not even sure where it came from, you know, because you bumped a chair or you did something. And what a bruise is basically a damaged blood vessel that's leaked out a little bit and is underneath the skin and it's visible. So what if you can't stop that from bleeding? And you just continue to bleed and bleed and bleed. And some people do that who have hemophilia. So if you have a patient ever who has hemophilia, you must be very careful with them because just bumping a foot on a wheelchair could be a problem, could be a real big problem. So hemostasis literally means to stop the bleeding. So it's promoted by reactions initiated by blood vessel injury. Constriction of the blood vessel will restrict blood flow to the area, and this is usually caused because of serotonin that's released. I don't know if you remember that from the last class. Platelet plugs are going to form, and then the plug and the surroundings are going to be infiltrated by something called fibrin. If you remember, I think last Thursday we had talked a little bit about the different protein fibers that you find in the plasma, and one of those was fibrin. Fibrinogen, which is an inactive form of fibrin. And we need to activate fibrinogen in order to get the clot made properly. So platelets generally, okay, so let me explain this diagram. It looks kind of bizarre. This looks like these are red blood cells, but they're not. These are supposed to be platelets here that are not active. This is the epithelial cells that line our blood vessel, and deep to that we have the connective tissue, and there's collagen fibers in the connective tissue matrix. And so generally speaking, when the endothelial lining is not damaged, prostacyclin is produced and nitric oxide, which actually is a potent vasodilator. Nitric oxide is found actually in Viagra. Like I said, Viagra was used for blood pressure medicine to lower blood pressure initially when it was first created, but then it was for erectile dysfunction. And nitric oxide is found in there, and it helps to dilate the blood vessels, what it does. So prostacyclin and nitric oxide are going to be present, released from the epithelial cells to prevent the platelets from sticking to them. So it keeps clots from forming, and again, nitric oxide is a potent vasodilator. Okay, so now what happens if we damage the endothelium? That's what we're seeing here. Now we've cut ourselves, and the blood wants to leak out, and we don't want that to happen. We need to put a stopper in the dam, you know, that has a leak in it. We have to stop it. So what happens is the platelets are going to be attracted to the collagen that's in the connective tissue, and they become active. They bind to the exposed collagen by something that's kind of like, I think of it like double-sided tape. It's called von Willebrand factor. Von von Willebrand factor will stick to the platelet and stick to the collagen, so it basically holds it in place. And then when this happens, it increases the bond of the platelets to the collagen, and then the platelets that are there in a positive feedback fashion are going to secrete some chemicals. These chemicals are ADP, serotonin, and thromboxane A2. And so releasing these chemicals will trigger other platelets to come to the site where the plug is being produced, and they also will release these chemicals, which will cause other platelets to come, again, in a positive feedback fashion. So what do these chemicals do? Well, serotonin and thromboxane A2 are going to constrict the blood vessel, because this way we slow how much blood is going to the area of damage so we don't hemorrhage as quickly. ADP and thromboxane A2 are going to cause other platelets to become sticky and attach and undergo the platelet release reaction, which we just described. And this continues until we get a nice little plug that stops the bleeding from going on. But we're not finished yet. Just because we have a platelet plug doesn't mean we have a good clot. We need fibrin. And so you need to get to fibrin if you're going to have proper clotting. So people who have hemophilia might have platelets and they might be able to get, the platelet plug produced, but if they can't get to fibrin, you're not going to have a functional clot. So the platelet plug becomes infiltrated by the meshwork of fibrin, and then the clot will contain platelets, and then all these little protein fibers and red blood cells that are sort of trapped, and it's a big mess. But that's why if you've ever seen a clot, they are red, because red blood cells are found in there as well. The platelet plug then will undergo a contraction and form a far more compact plug. So they'll get rid of all of the liquid that's found in the clot and make a nice hard structure. So the question is, how do we get from fibrinogen, which is inactive protein, to fibrin? Well, it actually occurs by a cascade of events that involves chemicals called clotting factors, and you need to make all of them in order to be able to get to fibrin. And there are two pathways, and what I'm going to do is I'm going to skip this slide. It's narrative of what I want to explain, and just go to this slide. Okay, so first of all, there are two pathways which can lead to fibrin. We have what's called the extrinsic pathway. This is where you have something from outside the blood that triggers the clotting cascade. So for example, if somebody had like if you put glass and blood together, right? The glass doesn't have heparin on it, it's just glass and blood. You're gonna get a clot. What triggers that clotting is the fact that there is an external factor that triggers this cascade of events to occur. In the tissues, if you've damaged, if you've damaged a blood vessel, within the tissues, you have something called tissue thromboplastin that's activated. And again, that comes from outside the blood. So that's why it's called the extrinsic pathway. The intrinsic pathway, I'm sorry, I told you about blast. The blast is on the other pathway. I apologize. No, the extrinsic pathway is just from the tissue. So tissue thromboplastin starts the clotting cascade. Intrinsic pathway, you have something within the blood that starts the triggering. So, okay, on a glass slide, if you put blood on there and it clots, it's doing that because something within the blood is causing the clotting to occur. Does that make sense? Does anybody have any questions about that?
So something in the blood is causing the
Yeah.
within the common pathway?
No, the intrinsic pathway. That's what it means. Intrinsic is from within the blood that the clotting cascade starts. Extrinsic is from without the blood, so outside the blood that triggers it. And then ultimately both pathways will end up in this common pathway. And this is what I want you to know. You don't really have to know all of the other, because the clotting cascade, the factors, the order of appearance of the clotting factors is not going to correspond with, like, the number is not going to correspond with when they occur. Actually, their numbers correspond with when they were discovered. So it's confusing. So you don't have to know all of those clot, but you do need to know the common pathway. That you should know. Now, ultimately, if we have the intrinsic or the extrinsic pathway, it doesn't matter. We're going to get to factor 10. That's X, right? Factor 10 is going to be activated. That will activate prothrombin, which is an inactive form of thrombin. It'll activate prothrombin to thrombin, and thrombin is going to cut fibrinogen into little fibrin threads. And now we have our polymer, which can complete our clots. So factor 10 is activated. That activates prothrombin to thrombin, which activates fibrinogen to fibrin, and we get our polymer, which is just a group of proteins all together in this clot complex.
So the common pathways, basically what you gotta know is within there it activates prothrombin, which goes to thrombin.
Factor 10, start with factor 10. Factor 10 activates prothrombin to thrombin. Thrombin activates fibrinogen to fibrin.
Okay.
Yeah. So we're gonna see, I think it's the next slide, that people who have hemophilia A generally do not make clotting factor 8. So clotting factor 8 is important over here in the intrinsic pathway, but not the extrinsic pathway. Notice that. People with hemophilia B, also known as Christmas disease, because it was named for Stephen Christmas. I don't know if he discovered it or if he had it. I'm not sure which, I forget. But clotting factor 9 is deficient. And if you look, once again, clotting factor 9 is used here in the intrinsic pathway, not the extrinsic pathway. So a lot of times people are like, well, I mean, why don't they just use the extrinsic pathway then to make clots in people who have hemophilia A or B? And the reason is because, remember that the extrinsic pathway is, there's some trigger, it's in the tissues, tissue thromboplastin, that activates the clotting cascade, right? Mm-hmm. But see, after you get enough factor 10 produced, the blood takes over and the intrinsic pathway is going to take over. So even if you start with the extrinsic pathway, you're going to ultimately default into the intrinsic pathway, and if it's not working, you're not going to get enough fibrin. That's basically it. Do you have a question? Also, you did this.
If the intrinsic doesn't work, you won't get enough fibrin.
Right.
Fibrin.
Exactly, and you need all the clotting factors to be able to get that to work. So, do you have any other questions about this? Clotting?
The intrinsic pathway.
Mm-hmm.
So first you start with extrinsic, which is something that causes the clotting outside of the
You
blood.
can start with the intrinsic pathway too. I'm just simply saying that if somebody has hemophilia A or B, they don't make clotting factor 8 or 9, which is exclusively used in the intrinsic pathway. People will get confused and say, well, why don't they just use the extrinsic pathway? And it's okay. And the reason is because it doesn't matter, even if you start in the extrinsic pathway, you're going to ultimately default to the intrinsic, because you start out outside the blood, but ultimately the blood takes over its own clotting.
Okay.
Does that make sense? So you need to have the intrinsic pathway working, otherwise you'll be in trouble. Okay, so now, as I mentioned before, there's a nice table here that just shows us all of these different clotting factors. And I mentioned factor 8 and factor 9. Factor 8 is known as anti-hemophiliac factor, and clotting factor 9 is known as Christmas factor, because again, it's you know, I think it was, I think he discovered it. I'm pretty sure he discovered it. But anyway, yeah, so that's hemophilia A, clotting factor 8, hemophilia B, clotting factor 9. Okay, so now we have a clot and the damage has been taken care of, everything's okay, now we need to get rid of it. So how do we get rid of it? We get rid of it when we activate a chemical called kallikrein. Kallikrein will convert a different inactive protein, plasminogen, to plasmin. Plasmin is able to eat at the clot and gets rid of it. So, people don't want me to write on the board because there's like hardly any chalk. So I don't know if you've ever heard of this chemical, TPA. Anybody ever heard of that? I'm gonna say no because it's quiet behind me. Okay. Anybody have any idea? We're talking about dissolution of clots. What do you think that TPA might be used for? Somebody has a particular pathology.
I don't know, removing the blood clot?
Yes, removing a blood clot, like in the case of what? What can they cause?
A stroke?
Yes. Now, if you go to the hospital when you just start to have it, and this actually was developed, I think, in the 80s, sometime in the 80s. If they're not sure, I mean, generally speaking, they like to do a scan, because strokes can be a couple of different ones. You can have one that's a clot, or you can have one that's hemorrhagic, okay, where you're getting bleeding. So it could be either. But if you have one that's a clot, they can administer TPA, tissue plasminogen activator, in an effort to try to get rid of the clot and reduce the damage. Yes.
Is that what they refer to as a clot buster?
Haven't heard that, but yeah, you suppose it would be. It would be, because that's exactly what it does.
Basically used like to remove the clot that can cause a stroke then, the damage?
That caused it, yeah.
Okay.
Yeah. Now, basically, what we've got is you've got plasminogen in your tissues already naturally, but in a pathological situation like that, it's not working properly. So what TPA does is it artificially stimulates the tissues to convert plasminogen to plasmin. That's what it does, and it goes after whatever clot there is. That makes sense? Okay. And lastly, anticoagulants. So we talked a little bit about heparin before, as I was telling you about, you know, whenever you do some of these blood tests, you have to put the blood into a tube that has heparin, because we know that if you have blood on glass, it'll clot, but if you have an anticoagulant covering that glass, it won't. So anticoagulants, they work in different ways, but essentially each one, how it works, it disrupts some portion of the clotting cascade. That's its job. So clotting can be prevented by calcium chelators. A chelator is something which acts like a sponge, and it kind of accumulates, in this case, all the calcium. So sodium citrate or EDTA are examples of anticoagulants that work this way. So calcium chelator, if we look back, you look at the different parts of the clotting cascade. Here we need this complex, we need calcium here. We need calcium in the common pathway. You need calcium in every pathway. Calcium is important. So if you if you have a calcium chelator, that's gonna prevent all aspects of the clotting cascade. Heparin activates antithrombin 3. Antithrombin 3 blocks thrombin. So we know that thrombin is important for doing what? What does thrombin do?
Makes hydrogen?
It basically cuts up fibrinogen into fibrin threads, right? So if we block thrombin, then we can't form fibrin and we can't clot. And coumarin, coumadin, warfarin, these are all forms of anticoagulants that block clotting by inhibiting activation of vitamin K. Vitamin K is something which is actually important for producing the clotting factors in the first place. And any foods that have vitamin K are going to be the things that are like bright colored foods, like squashes, like pumpkin, or like kale or spinach, those types of things. And this is why they say a lot of times, like, as you get older, you probably need to eat a little bit less vitamin K because of the fact that it can be a little bit detrimental. Yeah. So, you have a question?
I'm just wondering, why does coumarin, warfarin block clotting?
Coumadin, because it inhibits the vitamin K.
Okay.
That's how it does it, and then you can't make the clotting factors. And that's it. So I told you it was a quick little bit of a lecture today. And the last thing that we need to do is work on our white blood cell count, and then we're finished. And then on Tuesday, next week, we get to have fun with the blood typing game and doing our blood typing activity. We'll have just a little short lecture on blood typing, and then we'll get into everything else. I like the lab on Tuesday. I think we will too.
We don't have a quiz in here on Tuesday?
Yes, we do. Remember, red and white blood cell
Okay.
labs.
Just wondering on that quiz, is it gonna be like, you're gonna ask them normal readings or normal levels and what
Well,
does it consist of?
I could. So here's what I would do to study for the quiz. I would study your lab printouts that you had for labs 3 and 4,
Okay.
and study your levels, the correct units, the stuff on anemias, polycythemias, and then the white blood cell stuff. Again, study your printouts, and also study the PowerPoint notes. And anything I wrote on the board, like for example, I wrote that negative feedback loop up there, when we talked about red blood cells, remember?
Mhm.
Like, what triggers the EPO release and all of that. So study those things also for that. Do you have a question?
Are we having a test on that?
No, that's not until the test, that's on the test itself.
Oh, it's on that, okay.
Yeah, no, you can practice doing those things, but it's gonna be on the practical part of your midterm. Yeah, I won't ask you to do that for a quiz.
Oh, so easy.
You've been practicing. All right, so let's see. So on your desks, Kim put out some posters for you that you might want to refer to. These are going to be under higher magnification, like I said, than what you're going to be used to, and what you're going to be able to do with your microscopes. But that would be a nice reference for you, because you'll be able to see, like, as you look at a cell, if you're classifying it correctly. Remember, I told you that to do a differential white blood cell count, you need to count 100 white blood cells. And you use your clickers, the counters that you have here. If you see a neutrophil that's under the poly, you'll just click, 1. Then if you see a monocyte, click 1. If you see another neutrophil, click another one, that'll be 2. And you do that until you get to 100. And then what you can do is you can write your numbers down on the third page of your lab printout, or I'm sorry, the second page, where this table is, which shows you the normal percentages of each type of cell, and you can see what your percentage is. And you can kind of look to see if you see an elevated number of a particular type of cell, you can look at your specific white blood cell number indications and figure out maybe this person has this or that or whatever. Because all of these blood slides, they're from people and they could have problems. You know, they could be sick or they could have, you know, mono, something like that. You never know. So the question is then, okay, and again, you're going to use your blood smear slide. It's in your box. Question is, how do you know which cell is which? So this one I'm going to attempt to show you here. Now, I might not be able to find, for example, basophil. I'm going to look really hard for one, but basophils are very rare. Okay, so if you remember from the last class, we were talking, there's 5 different types of white blood cells. 3 of them have granules and 3 of them do not. I'm sorry, 2 of them do not. Alright, so in this slide, we can see 2 granular leukocytes here and here. These are the most abundant white blood cells. So these are going to be neutrophils. Neutrophils have the nucleus that has a bunch of different like little lobes to it, and it always looks a little funky. So it's really easy to see those, to point them out. The cytoplasm does have granules, but they're not really that visible. It's mainly the nucleus that tips you off. You're looking at a neutrophil. So they're the most abundant. Then over here, this cell, which is almost all nucleus and very little cytoplasm, it's not super big, that is going to be a lymphocyte, which you can see on the poster. Lymphocytes are the next most common type of white blood cell that you'll find. Remember how you can remember the order, never let monkeys eat bananas? Remember I told you that? So neutrophils and lymphocytes, and boom, those are the first 2 that I found. The other ones might be a little bit harder. But I'm going to try to find a monocyte next. Here's one. Now, monocytes, I mentioned this to you before, they usually don't appear round because they are going to be on the move a lot. Remember I said when we looked at the video in class, it's the cell that kind of slinks through into the tissue spaces. Monocytes become macrophages out in the tissue spaces. So when you see them, they are pretty big cells, but a lot of times they're not going to be perfectly round, like this one right here. You can see the nucleus is very big. You've got more cytoplasm here though. If we compare that to the lymphocyte that we saw previously. It escapes me. There. See, the lymphocyte is a lot more nucleus, and it's not as big. Monocytes are very big. Okay, now comes the hard part. Eosinophils and basophils. If I can find an eosinophil, which you generally can find those more readily than you can find a basophil. But I'm searching. In the interim, I found this one. What do you think this one is? Yeah. This is a lymphocyte. It's all nucleus, very little cytoplasm. So it is a lymphocyte. Like I said, monocytes are usually bigger, you see more cytoplasm, they're usually not round. I need to find- Even under scanning magnification, you can usually find an eosinophil pretty well because it glows kind of red. What you're looking for is the nucleus is always gonna be blue, but you're looking for granules that are around the nucleus that are pretty much the same color as the red blood cells, because eosinophils appear red, pretty red. I actually found a basophil. This is not common. This is very rare. Basophils. Yeah, more than likely, you're not gonna see this. Basophils have, see, the blue nucleus is there, but there's little dark purple granules that are kind of scattered throughout there. That's a basophil. But again, it's not very common. I think on this TV screen you can see it better. It's like little dark purple specks, and the nucleus is in there behind it. But can you see that?
Looks like it.
Yep.
Huh?
It looks like it.
No, neutrophils definitely don't look like that.
Is the
I
one-
can show you another neutrophil.
Is the one on the top right a monocyte? Or
Top
what is it?
right.
Up there on the left.
Here,
Oh,
top
No,
left.
the top right, it's pretty dim.
Oh, this?
Yeah.
No, I think that's artifact. It'll be a little bit more obvious. No, neutrophil, the nucleus looks different. This is where you need to really look at the characteristics of the cells, because like I said, here, when you're looking at this, you see these little dark blue specks. You're not gonna see that with a neutrophil. Neutrophil, you're gonna have the dark purple nucleus that's like in lobes that look, strange. I'll see if I can find another neutrophil and you can compare.
You said that's an artifact?
Yeah, you see that sometimes. I'm gonna try to show you another neutrophil so that you get, since that image is in your head. So, neutrophil. See, these would be neutrophils. See how the nucleus is like all different shapes? And then this here would be a lymphocyte, another lymphocyte.
Can you show another eosinophil?
Well, I'm trying to find one. I haven't found an eosinophil yet. Unfortunately, I don't know if I'm gonna be able to. Doesn't really look like it has. Like I said, you can use your oil immersion lens, also, and it can help you to see it a little bit better. Okay, here, this is a lot, more obvious as a neutrophil. Here. Yeah, here's a good one. Neutrophil lymphocyte. You see that? Like I said, use the posters too for reference, they'll help you out. And I need to try to find an eosinophil. Tell you what, you guys can go ahead and grab your microscopes and start getting set up. And I'll just keep searching